This is the Convergent Science Network podcast. Leading researchers in the domain of neuroscience, brain theory, and technology are interviewed by Paul Bechir and Tony Prescott.
This is Paul Bechir with the Convergent Science Network podcast, and we're here at the 10th anniversary summer school, the Barcelona Cognition, Brain and Technology Summer School. And I'm here with Lynn Uhrig and Bechir Jarraya, who are presenting this morning their collaborative work on anesthesia, the brain, the thalamocortical system, and also how we can develop animal models of this. So now, Lynn, how do you think the kinds of systems you're looking at are helping us understand how the brain works? So the particular angle that you take of anesthesia, how is this really helping and how is it strategic?
I think it's very important to know what we are doing in everyday clinical work because so I'm an anesthesiologist at the beginning, and when I started my residency in anesthesia, the first question I asked was at one moment how anesthesia works on the brain. And the answer I got at the very beginning was, okay, it works. Why do you ask this question? So that's why where came the idea to study anesthesia, because at some moment the problem of anesthesia was to make it safe for patients. So there was a lot of improvement in the hemodynamics of anesthesia, in the respiratory field of anesthesia and so on, but not really in the neuroscience fields of anesthesia. So it was like at some moment you can get a shutdown of the brain. Okay, it's nice. People don't remember what happens, but then you don't. At the beginning when I started my residency, there was not much more that was known about anesthesia, and so at some moment was like I want to know better what I'm doing, to take at some moment perhaps better care of the patients I have in charge.
Mm-hmm. So it's fair to say that actually we don't really know why anesthesia works.
No, I think today we know more than 10 or 15 years ago. Okay, but still today we don't really know why people lose consciousness with different drugs we use in everyday life.
And so what was really impressive in also your talk is the overview you gave of the different, let's say, interventions we have available today to induce these states of consciousness that you might use in the surgery ward. So what are the most dominant interventions that you're using?
I think between the most dominant drugs we use in everyday clinical practice, there's propofol. Propofol is used everywhere in the operating room. Also, for the volatile agents, is most sevoflurane, sometimes isoflurane, what is more older drug. And then if you have patients in the intensive care unit that are not very stable, we use ketamine because it doesn't act on the hemodynamics of the patients. So I think these three are mostly used, and probably in the future what will be used, at least in the intensive care unit, probably dexmedetomidine, because it's thought to induce a sleep-like state. What would perhaps be more physiological, a condition for the patient.
But that's right now more in experimental phase, so that's already being used?
It's already being used in intensive care units, yes. When you wake up patients at some moment to induce like a sleep-like state. Yes.
Right. But now the pathway of action of propofol and ketamine, and this also will be relevant later when we look at you, understand you and analyze your data, is rather opposite.
Yes, yeah, it's completely the opposite on the brain.
So we can say propofol act essentially driving the inhibitory system.
Yes,
right. This is fair to say, in a rather non-specific way.
Yes.
Okay. Well, ketamine is more specifically acting on the excitation on the inhibitory system.
Yes.
Right, because it acts on the NMDA receptors on the GABAergic cells. But is there some sort of regional specificity to that in any way?
Let's say for the ketamine drugs, it's mainly acting on the cortex, not on the subcortical regions, and on regions like the hippocampus, the amygdala of the hippocampus. And for propofol, it's thought that at the beginning it acts on the cortex and in second phase of action, it acts on the subcortical regions. There are human studies, one human study by the UNLVD who showed this like 10 years ago.
Mm-hmm. Right.
In patients.
So this is already one mystery that we should try to clarify, right? Yeah. We have these two drugs. They in some have an opposite effect on the inhibitory system of the cortex, while still leading to the same outcome, with loss of consciousness. So we have to solve that one before this podcast is over. And this is also what you have been trying to do, following very specifically a very unique paradigm that has been pioneered in the lab where you both are working, which is a local and global paradigm, right? So I don't know which of you two would like to speak for the local global paradigm, but Bechir, you have been very active in that. So what's so interesting or unique about this local global paradigm?
The local global paradigm is a series of sounds that you can listen to. This is what we call a paradigm. It was the idea came in from our friends and colleagues Stanislas Dehaene and Lionel Nakache. What does it mean? Local versus global. So if you listen to small sounds like beep, 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 boop, you have same sound, identical sound coming in, and then what you call a deviant. So you violate a rule at the local level because it is a small, tiny scale of time. But this is have been investigated for decades, and if you do EEG, called ERP, Nathaniel described what we called famously the mismatch negativity. People were excited about it, tried to perform this paradigm in anesthetized people, in people emerging from coma, but unfortunately was not discriminative of loss of consciousness. So even without consciousness, your brain, you can still process this mismatch negativity or the local part of this local global paradigm. So the trick there was to multiply the sequence, this local sequence, several times, and this time introduce a violating sequence. So the new sequence that comes by the end, like for example, beep, beep, boop, beep, boop, beep, beep. This last sequence is violating the other one. This is the global violation. And it turns out that it works fantastically because you really need to be conscious to process this global effect and that to have your brain realizing this sequence violation. If you have, for example, a vegetative state, which is a patient that never recover any consciousness, then you definitely cannot realize this global part of it.
Mm-hmm. And do you think you can stack these sequences indefinitely? Is it like recursive?
Currently, the structure of the sequence by itself is not aimed at recursive detection. This is definitely there are variety. We are developing the varieties of this paradigm to check for syntax, for numerosity. We did that even in animals, but so far we consider that as a marker of conscious access.
Mm-hmm. So now we have this paradigm where in some way we have local and we have global deviations, right? And then, so you see this as a specific probe of states of consciousness or aspects of consciousness.
The global part of it, as opposed to the local. This was demonstrated by Tristan Beckenstein, a paper with Lionel Nakache and Stanislas Dehaene, and they showed that in humans, at least, healthy people distinguish very nicely the global effect from the local effect, meaning that the global effect will need a large-scale cortical activation, will induce large-scale activation. Actually, our contribution came in with the animal aspect of it. Mm-hmm. Because the question was, in that time, there were two questions. First, could we infer the same thing in non-human animals and our closest cousins, let's say non-human primates? And second, starting from an animal model in which we can induce anesthesia could we manipulate this global detection with the manipulation of consciousness?
Right. Okay. So if we now look at this, the human case, what we look at is now a paradigm that allows you to access, if you want, informational aspects of conscious processing. So we're not saying much about the experiential, phenomenological aspects of consciousness, right? This is not part of the discussion now.
Exactly.
Yeah. So we really focus on access consciousness. And then what you see is a very distinct correlation between, let's say, these local sequences and, let's say, A1 processing in the temporal lobe, or more broad activation in what in your center is called the global neural workspace.
Absolutely.
Right? So what are then the core nodes in that global workspace that you observe in this paradigm?
So if the regions we activate with this local global paradigm, and especially for this global effect, there are parts of the prefrontal cortex, there are parts of the parietal cortex, there is the anterior cingulate cortex and the posterior cingulate cortex, at least for the cortical regions that we activate with this paradigm. If you go to deeper structures, there are structures of the thalamus, like the parafascicle nuclei of the thalamus, and part of the striatum that you can activate with this paradigm.
So now we have a starting point, right? So then also you say, look, there's a form of conscious processing we can now manipulate. We have a neural substrate, and then indeed the big step, as you also earlier said, Bechir, the big step was now to bring that to an animal model, because then you would have more experimental control, right? So how well did that generalization to the animal model really work, Lynn?
I think it worked quite well, because at the beginning we had no idea if the animal could detect this global effect. What was quite sure that it could detect the local effect, because there were older studies with electrophysiology that showed that if you do invasive electrophysiological studies in the auditory cortex, that you can get the mismatch negativity that you can record in the auditory cortex. So we that at least what we expected. Then was the question, could you detect something more complex? Because if you want, yes, to learn like a rule, like if you have like 5 identical sounds, then you get this as a rule, and at some moment you get this global deviant. And in all the animals that were tested, they were tested at several times in the scanner. There were 3 animals in total, because for monkeys often it's difficult to have more animals because you have to be trained at the beginning. And the data we show were group analysis of this monkey. But if you look at the individual level, all the monkeys could get activations in the prefrontal cortex, the parietal cortex, anterior cingulate, posterior cingulate. And some also had activations in the hippocampus.
Mm-hmm. But now, what's the magnitude of these differences between the local and the global variations of the task, and how does this magnitude compare to what you observe in humans?
There is actually 2 remarks there. First, there is a strong hierarchy. So the local global paradigm introduces the notion of hierarchical levels of violations, of sequence violations. And that is really important. This is in line with what we call the predictive coding theory. And what we saw already in human, and we see exactly the same homology in monkeys, is that because the global violation is hierarchically superior to the local violation, it also activates cortically a high cortically organized frontoparietal network, whereas the local effect was really activating the, let's say, low-level auditory pathway, including within the brainstem until A1. And we saw that exactly in a homologous way in the macaque, meaning that in the macaque, we saw the auditory pathway from the brainstem nuclei until A1 for this low-level deviant, which is a local effect, while when we go to the global violation, the second level of hierarchy, you see you see involvement of both prefrontal and parietal cortex. And we went even a little bit further to see how much this is really specific to the global effect by doing a technique in fMRI data called psychophysical interaction, PPI. And this put in a seed in A1. PPI could show that really global effect increased specifically, brain activities increased specifically in these frontoparietal singular areas when the global deviant comes to the macaque auditory system.
So in the human case, we see some a frontal parietal signature of the global task, right? And then in the macaque, you see something similar, also in fMRI, right?
Exactly.
We're doing fMRI in the macaque. But now we have, of course, this whole conundrum of homologs, right? What are homologs between macaque brain and human brain? And actually, if you just look at the pictures that you present, also in your work, it would appear that, for instance, these spots of activity are a bit more lateral in the macaque case as compared to the human case. Maybe I misinterpret, but how do you deal with this issue of homology?
It's really a tough issue. I like the question because we are interested in that in brain monkey comparisons, because this shed light eventually on the uniqueness of human brain, of course. And technically, it remains very, very challenging to do direct homology studies between the anatomy of the primate brain and the human brain. There are already some tools existing in the literature. There are still a lot of work to do there. Mm-hmm. And we are interested to do that in the future. Right. Because it's tough to compare brain maps by direct visual, our visual system, basically. And the notion of homology is not that trivial that we could think. What was important in our case is really to see that the two orders of violations, two hierarchical orders of violations in monkeys, just like in humans, were paralleled by two hierarchical order of cortical activations. That was important in our case.
To see that. But now imagine we do something stupid, which I like, because these are things I'm good at, and we just inflate or sort of in a very linear way map this macaque brain to a human brain. Would you predict that then the nodes in this global network in the macaque would align with those in the human brain or not?
I'm not sure we can really fix the problem this way. There are tools in Carrot, for example. Carrot software offers tools to do just this direct alignment by simple inflation. However, you need to keep in mind that the human development of a human brain was not just about inflation of volume, as you know, but also of specification and local specification, and on top of it, all the education and training. This is clearly one of the challenges open the next years, is how to develop really proper tools to do these direct comparisons, because with the ability to train monkeys, and especially macaque monkeys, to sit in the scanner and to listen to the exact same paradigms in the same conditions than the human counterparts, we start to, for a while already, with wonderful Nikos Dogothitas and Doris Tsao and other people, we start to have this unique opportunity. I have two maps for exactly the same experimental setup. How can I deal to have a direct comparison, a computational comparison? That's something we are also working on.
But what is interesting is that it might mean that the midline structures of the macaque brain might be organized somewhat differently as compared to those of the human brain, even though, let's say, from a phylogenetic perspective, they're relatively close. Would you agree with that?
Again, I would say that not only comparative anatomy is needed there, but we need to automatize tools based on tracer studies, for example, to match the two brains. Will there be clear transform between the two brains. If we think about all the millions and years of evolution that made this strong job, I'm not quite sure that we have an direct easy transform.
Mm-hmm. Right. Okay. But then, so we have this paradigm. We see that we go from a local process to a more global process. Okay. But now what you're relying on is a paradigm that assumes that the animal is basically passive but aware, or as the human is passive and aware in this resting state paradigm. And it's not that the resting state was invented because its behavior is so interesting. It's just the simplest thing that people can do as they sit in a scanner or lay in a scanner, right, surrounded with this noisy equipment. So now we're forced to do it with the monkey as well. So that means, and also I brought this up in the talk, right, that the monkey is sitting there. We assume the monkey is sort of at rest and aware, but we have actually no real idea whether this is in any way pertaining to the consciousness of this macaque monkey, right? So how do you see this as a limitation of these studies, that the monkey, that we have no way, that there's no reportability, that there's no overt behavior coming out of this monkey? Do you see this as a limitation, or you don't think this is a problem?
It is, of course, a limitation, very clearly. So how to fix the problem? One way is to train massively the monkeys to report for the detection of the local violation versus the global violation, of course. We thought about that. The problem with that is that it implies a lot of training with the paradigm. And in our case, we tried to, at least this was our strategy. We discussed a lot before starting this research program. We tried to keep really the monkeys naive to the paradigm, because overtraining with the paradigm is always a confounding source for the interpretation. Now, I agree. You pronounce the word reportability. But I would say also that we start to accept fMRI as a reportability now in humans with disorders of consciousness. When you see all this work by the Liège and Cambridge groups showing with this tennis imagery paradigm in highly disabled people with minimal conscious state, actually these people really do not report except through fMRI. So fMRI activation starts to be an acceptable reportability, let's say.
Well, but that's maybe more a message of hope, right? Because it doesn't work in all patients. Right?
No, it doesn't.
So, but okay. So here we have our paradigm. We have a local global effect. It's sort of, let's call it weakly analog to what you see in the human. I mean with weakly because we cannot really nail whether it's homologs or not. But you see a local global effect involving frontal and parietal systems. So fantastic, right? Great. So now we have calibrated, we mapped the paradigm to the macaque monkey. But now the real stuff starts, because now you start to manipulate these states of consciousness with propofol and ketamine and other drugs. So Lynn, what happened when you started to do that?
So at the very beginning, we had to make sure that the anesthesia protocol would work, because you cannot like this put your monkey in the scanner, put one drug, and then see what's happened. There was a lot of work that was done outside the scanner before going to the scanner to make sure that already the anesthesia level was stable during the whole experiments. So the anesthesia model was based like for propofol, it was, and also for ketamine after. It was based on behavioral scale of the monkey, because you want to go to general anesthesia. So normally general anesthesia, you are not conscious. And then we had like behavioral test that we did in the monkey. And also, you had to make sure that you have a stable EEG pattern on for your anesthesia, because you cannot say that you use like X milligram per kilo of a drug and then it will work for everybody. Of course, it didn't work for the monkeys, so we had to adapt the anesthesia concentrations on the behavior scale and on the EEG before doing the whole paradigm after in the scanner.
And then, okay, first you have to fine tune the paradigm. You have to get to the right dosage. So you sort of nailed that problem. Of course, it's a very hard one, right? But then in something you saw a differential effect of the ketamine and the propofol was not identical in its impact, right?
No, it was like really closely for the local effect was quite close, but let's say we weren't really interested in this local effect because there's a lot of literature showing that if you do electrophysiology, even in humans, that if you go to general anesthesia, often you lose some mismatch negativity. So we're not surprised that at some moment you don't get a local effect in these animals. What was quite surprising was for the global effect, because it could have been that, okay, you have no local effect, you will have no global effect at all, and so you have nothing. You will activate your auditory system when we present all the sounds or do the contrast for all the sounds for fMRI. That was at least what we were expecting, because it is reported in literature that the sensory cortexes are still active under anesthesia, but not beyond. And then at the beginning, we were quite surprised that we had no global effect at all for ketamine, and that still we had activations, especially in the prefrontal cortex of propofol. Mm-hmm. So that was one explanation, could be that they act on different receptors, and that what gives these explanations. But it's not completely sure. We cannot completely make sure that is.
No, but so the main thing that you observed, that you also described, is that with ketamine, you don't see the activation of this frontal parietal network, yes. Right? Well, with propofol, you do. But now the propofol case, is this frontal parietal network then still identical to what you might see in the control case?
No. When you compare it to the wake state, what you see that you still get activations in the prefrontal cortex, but activations you never get is in the parietal cortex. Mm-hmm. So whatever condition you test, whatever analysis you do, you all the time with propofol, you block the activations of this global effect in the parietal cortex. You can get it in the auditory system, you can get it in prefrontal regions, but never in the parietal cortex. So
is that a significant observation in your mind, that it is in particular affecting the parietal part of this frontal parietal network?
I think it's quite significant, because there is like a lot of literature showing that you have a prefrontal parietal deconnection with anesthesia. There are a lot of work done by George Mashur in the US, showing that you have really deconnection with all the anesthetics, whatever the anesthetics you use, like propofol. He showed it with ketamine, with sevoflurane on EEG data, and he shows that all the time you get a deconnection between the prefrontal cortex and.
But you show a bit more, right? It's not disconnection; it's disappearance.
Yeah, it's disappearance of activation.
It's actually more extreme than that, right?
Yeah. It's a disappearance of activation.
Mm-hmm.
Yeah.
So, but that would suggest, and it's also maybe something we can get back to later, that this parietal part of the frontal parietal is maybe much more of a hub in that system than the frontal part. Yeah. Would you agree with that? That's a working hypothesis?
Yeah.
Okay. So okay. So we have this differential effect of ketamine and propofol, and then you started to analyze in much more detail, which is really very interesting, the specific organization that responds, right? You start to look at the effective connectivity. How is the effective connectivity then changed due to propofol in the context of this task? So because then that you could use that then again as also a marker for reinstating this excess consciousness, right? This is where we're going with that. So how useful was that analysis? We look at this effective connectivity and how it is then changed by these different drugs. What did you observe there?
Interestingly, if you want to study anesthesia or even consciousness, there are basically two different ways. There's a way where you challenge the brain with a task, with auditory stimuli. It's even easier in people with eyes shut down. As Lynn mentioned, people should really be aware of that. Under anesthesia, obviously there's no shutdown of the brain. There's still a strong brain processing of a lot of information. Does it mean that there is conscious access? Does it mean that there's a memorization of these data processing? But there's at least, for example, with the sound we display and with fMRI, we see very strong activation of all the auditory pathway. Very strong. Mm-hmm. So I said a word to my colleagues, surgeons, be careful of what you are talking during operations, because patient is listening. Mm-hmm. Well, now another way is to study the spontaneous fluctuations of brain activity, so-called resting state. Very, very extended field of neuroscience now, since the pioneer work of Marcus Raichle in St. Louis. And if you look at these spontaneous fluctuations, there have been work under anesthesia, but here we introduced a new way of analysis of these resting state networks called dynamic resting states. What does it mean? It means that people who are familiar with resting states know that you scan your subject or your animal for 10 or 20 minutes without specific task, and then you analyze what we call functional correlations. I would prefer the word functional correlations over functional connectivity because it's more objective, but still. And you will describe networks that we call, for example, default mode network, attention network, that will pops out very easily through some code. And here you're dealing with the whole picture of your 10 or 20 minutes of acquisition at once, while we know that even the functional correlation between two areas fluctuates during all the session of acquisition. Sometimes could be stable. So here comes what we call dynamic resting state, because through a sliding window phenomena, it can also be done without the sliding window, you can cluster brain states. Here we used an unsupervised method called k-means, cluster all this resting state data into several brain states. And these brain states proved to be extremely useful. What happens there? So we could see that if we, let's say there's 7 brain states that explain your resting state configurations, and with the clustering of brain of your resting state, you will do the same clustering in the awake state, then under anesthesia, then propofol, ketamine, whatever the anesthetic, and with different dosages also of these drugs. Suddenly, we saw that there are brain states that are very close to the anatomical connectivity of the brain. And those states, we will call them rigid states, because at that moment, when your brain is in this state for, say, a few seconds, your resting state is 100% explained by your connectomics. You have no freedom. You are jailed by your structure, your brain anatomy. So you have high similarity between structure and function. And when it comes to the other part of the spectrum, we found a state that is completely the opposite. This is a configuration of resting state that is completely uncorrelated to the structure. And we called a flexible, it's very high, highly flexible brain state. It means that at some point, your brain jumps from a state to another, and these states have completely different properties. Now, the nice finding we made in the group is that when you are awake, most of the configuration of your brain can be explained by many brain states, and not only one, with a heavy shift to these flexible states. Mm-hmm. Whereas when you go to under anesthesia, propofol, ketamine, even others, you see the exact opposite. It means that your resting state is explained mainly by the rigid state, and it means that you have a strong influence of your connectomics to explain your spontaneous brain fluctuations. It means that these spontaneous brain fluctuations are not so spontaneous. They are shaped heavily by the structure.
Mhm.
So to summarize this description, let's say that we could say somehow that being conscious, meaning being completely free out of our brain structure.
Mm-hmm. Right. But the thing is, if you go to the brain structure itself, that's an estimate of a DTI.
Yes.
Right. And DTI is relatively incomplete in sort of assessing the structure of the brain.
Absolutely. That's why actually in our study, we used a database that is not a DTI based database. This is COCOMAC that summarize all the literature of tracing in macaques.
Mm-hmm.
And there's a strong literature of neuroanatomy. Sure. And so in our case, this the structure matrix were coming from these studies.
Okay, so then we have these 7 dynamical states in which you could cluster the resting state activity that you measured in the macaque brain. Then we see that in the low entropy case, and you also showed that let's say we're not conscious or we're close to sleep or we're under severe sedation, it's perfectly matched to the structure. And in the case of wakefulness, we are sort of have a much higher variability that cannot be related to the structure directly. But do you see then a very discrete transition in those 7 states? Like, is there a sharp threshold? Like, if I'm mildly sedated, I'm immediately below that threshold somewhere, or it's a more gradual transition?
It's a very good question. I think for the moment with the data we have, we cannot answer the question because when we did these experiments, the animals were either in one state or another state. We didn't do transition studies. But I think it's something very important. And we had a lot of discussions to do future experiments with transition states, so to going from a wake state, but very slowly to an anesthesia state, even during which can last for several minutes. So to go from one state to the other, and also the other way around to see what happens when you are in anesthesia and you wake up. But I think that's studies we have to do to explain, really to know if it's something you go from one conscious state to the unconscious state and you lose some of this brain states we described, or if there's a transition that could be slowly.
Mm-hmm. And okay, but then you also highlighted the fact that you see very specific kinds of correlations disappear under sedation, and then also it seems to be dependent on the kind of drug you are using, right? So we talk about the sort of the positive correlations or the negative correlations. So here we have this functional connectivity diagram. That means I look at the correlations across all the voxels that I'm looking at. So how is this then specifically affected by either ketamine or propofol?
What we saw for these correlation studies with propofol and with ketamine, especially when you look at the stationary connectivity, but also when you look at the brain states that's still present under anesthesia, that you mostly use, that you have a loss of mostly the negative correlations in the brain. There's still a lot of positive correlations that are still present in the brain. They are less compared to a wake state, but there are still many that are present. And what you lose completely, at least for these 2 drugs, are the negative correlations.
Do you find it surprising?
Not completely, because there is, I think, like one hypothesis that negative correlations could be important for information processing in the brain, and then when you're not conscious, you have no information processing, and then we lose them.
Right.
So that's one idea. That's why I was not completely surprised to find this.
But you could also argue that since you are manipulating the inhibitory system, right, that might have a specific effect on your negative correlation, because these are the guys who are sort of managing that part of the process.
Yeah.
Would that be reasonable as well, or you think that doesn't make sense?
No, that could be another explanation. I never thought I think of this one, but could be a good explanation too. Huh?
Okay. Let's try that. You tell me next time. But now the other thing is that then, which was really exciting, because you are coming from, both of you are fully committed to the global neuronal workspace idea. And so you then started to look at the nodes the frontal parietal nodes that would make up this global neuronal workspace, and you start to look at how they would be specifically affected by either propofol or ketamine. And then, in some sense, surprisingly, the effect was sort of like indistinguishable, like coupling across the global workspace nodes disappeared in both cases, sort of in a comparable way. And this is a fair summary, I think, no?
So if you want to link both findings, for example, with the task, with the local global task, and in the resting state, true that findings were more homogeneous with the resting state approach. So all anesthetics share the same signature, which is the loss of independence of resting state from brain structure. Whereas with the task, it was more subtle and more. There's a myriad of possibilities. But actually, there is an apparent paradox, and a way to match both is that the idea is that the integrity of this global neural workspace is really key. If you take a piece of it, then the whole system falls down. Mm-hmm. And it looks like, there was always a mystery. How can anesthetics that have completely different molecular targets, different neuronal population targets, different pharmacology, and post-receptor molecular events converge to the same result? I mean, it looks impossible in terms of pharmacology. And actually, one of the potential explanation could be is that because they deprive the global neural workspace in a different manner, partly, completely from its information processing. But it's enough to disorganize part of it to lose completely the conscious access.
Mm-hmm. Okay. But what I found surprising is that the effect is so non-specific, right? So here we have two pharmacological agents that have very different impact on neural circuits. And at this very macroscopic level, at which we analyze the system, the impact is actually indistinguishable, which worries me a bit, because from a global workspace perspective, it's an information processing view on the brain where you say, well, we have all these processors, they're competing for access into the conscious buffer, if you want, and they start to broadcast into this buffer when they are sufficiently driven. Right, but now, in some sense, here there's no signature really of such a buffer being maintained in any systematic way. It's there, it's not there.
It might be also, Paul, that the measure of resting state and its relation to structure is not really specific to the agent. It's just because you end up having a complete loss of consciousness. It's like a phenotypic biomarker, let's say. Mm-hmm. This is also a way to explain why when you do task, you have more subtle differences. And while when we do the resting state approach, it looks all the same.
Mm-hmm. But this is actually an important point, right? Because, for instance, in other studies of consciousness in humans, you might see very distinct kinds of subnetworks for consciousness that's more metacognitive, oriented towards the self, and conscious states that are more oriented towards the external world, right? So if you have actually the kind of paradigm you're using here, none of that is being controlled, right? So maybe you're not driving the system efficiently enough to actually see anything that is really specifically impacting then that excess consciousness that you're after.
Because again, something I like to say is that consciousness is an ambiguous word also. So you mentioned metacognition and self-monitoring, which is not necessarily what local global is probing. This is again another view of consciousness, of self-consciousness.
Right. So, but what I, even though maybe it didn't appear like that, but I was trying to support your point by basically that I would not be surprised if this sort of resting state paradigm is in the end not helping you. It's more pulling you away from where you want to be, because there's no specific the brain is not configured towards a certain goal. So do you also see that as a challenge?
I agree. And actually what goes also in this direction is that this signature with resting state similarity is now explored by other groups for other circumstances with loss of consciousness.
Mm-hmm.
The group of Oxford with sleep, for example, and at ICM, at Bechir with and Liège with people with disorders of consciousness. And they are very exciting data, finding again this same signature. And here we are beyond different pharmacological agents. We have different conditions, different even brain anatomy sometimes with lesions.
Right. Okay, but then so to close this part up, and then we go towards the thalamocortical system. The other thing that I find really interesting about the data you present, because it's so specific, is that since you're also in the global workspace tribe, in some sense, your data shows that we should take global a bit with a grain of salt, right? Because like in the macaque brain, we talk about 12 identified nodes, more or less, right? So how global, in your opinion, should we really think about this global neuronal workspace in the context of the task that and the kind of manipulations that you have been using?
The term global actually refers also to the global broadcasting of information, and actually there is no single paradigm that activates all the global neural workspace. So obviously, if you are assessing, for example, an auditory paradigm, you don't see visual activations, and vice versa. So the GNW theory should not be seen as just an anatomical, clear anatomical network as DMN, for example, or so forth, but as a general idea of cortical broadcasting of an information. And the fact that here the auditory information went much beyond when there was a global violation and went to prefrontal areas close to frontal eye fields, for example, to intraparietal sulcus with area VIP, shows this second level broadcasting of information. But it doesn't necessarily tell you that we have X nodes or Y nodes in specific species. This is the way I see it.
Well, but I understand, but you could argue that the global workspace in the end is talking about broadcasting it to some sort of buffer. You broadcast it to some sort of memory system, right? And then the question is that memory system what you now actually reveal in these paradigms, frontal parietal as a memory buffer in which these broadcasts land? Or is this frontal parietal system you visualize, let's say, an auditory specialization of a much larger kind of buffer in which you can potentially broadcast?
It's really impossible to answer definitely this question.
But if you would speculate, what's your hypothesis?
If I speculate, I would say that the apparent homology we see between the macaque response, the macaque cortical response, and the human cortical response, let me know that we are really having a description of between brackets, a macaque global neural workspace, I believe.
Mm-hmm. Okay. So we have a starting point now to start to look at the details of the mechanisms of anesthesia, right? And so this local global paradigm helps you in doing that. You revealed it now in the macaque. So now we have an animal model. So now we start to can become more specific in manipulating it. That's also what you do with your deep brain stimulation. Right. So why do you believe deep brain stimulation might help you to then manipulate these states of excess consciousness?
So just let me say a little bit the story and why we got into there. From a personal perspective, I'm a neurosurgeon, and when I started my residency in neurosurgery, end of the 90s, early 2000. I was surprised by a category of patients that have a strong, big head trauma or a catastrophic stroke, Many of them die, unfortunately, but some of them, and more, thanks to modern medicine, we are making these patients living, but unfortunately many of them in very bad conditions. And here it's not about motor recovery, but actually it's about consciousness recovery. It's an increasing issue in our modern world. I mean, think about it. 50 years ago, there was no such patients. It's like almost an invention of modern medicine, because these patients, until the 50s, with the appearance of the modern intensive care and surgical techniques, died in 100% of the cases. I mean, the stroke was so strong, the trauma was so ugly and dangerous. All of them died. So modern medicine is producing a new medical condition, which is a vegetative state and or minimal conscious state. And it's really intriguing, because once the phase issue, the acute phase, is done and they survive, they start opening eyes. Family are happy, but unfortunately it doesn't mean they are conscious. They are aware. They are awake. They have wakefulness, but not necessarily aware. And I was intrigued by these patients, because they could stay weeks, months in the department. Of course, rehab centers don't take them, and now they start to have specialized centers for these patients. So there is an issue. And at the same time, there are famous personalities. We think about Aaron Sharon, who passed away a few years ago. Currently, Michael Schumacher. So in the headlines, we see more and more these conditions of what we call disorders of consciousness. In the same time, deep brain stimulation, which is a surgical technique that consists of implanting electrodes inside the brain, became more and more fashionable and more effective in another disease called Parkinson's disease. And thanks to the work of Professor Ali Benabid in Grenoble, this make it very popular, and now there's something like 150,000 people in the world who are walking around you with these electrodes with Parkinson's disease or tremor. So because this revealed to be a powerful mean to recover from Parkinson's, one of the idea was, could we actually identify brain targets to do deep brain simulation to recover from consciousness loss. Of course, it's much more challenging, but because consciousness now becomes a neuroscience issue and not only a philosophical question, and now it's in the lab, in many labs in the world, it starts to be considered as a function, like motor function, like language. And because the patient lost this function, it might be that there are techniques to recover from that. So this is general context. And actually, there are already some few reports in literature back to some decades ago with some groups in the world who tried to do thalamic stimulation to make vegetative patients recover. Why thalamic? Actually, thalamus have a very strong connections, a very widespread, through large-scale cortical networks, frontal, prefrontal, parietal, singular cortex, precuneus, all these key areas for conscious treatment. So the idea was there. And we know also that thalamus is highly involved, because some strokes of the thalamus can, even small strokes inside the thalamus, can make consciousness disappear without any other neurological problems. That's how there's a convergence toward these DBS of thalamus to try to restore consciousness.
Mm-hmm. So, but then, how successful have you been so far in restoring consciousness with deep brain stimulation?
We, in our lab, we are doing preclinical work We are not doing a clinical trial, but we are using exactly the models that have been developed by Lynn using the anesthesia. Of course, anesthesia is not the perfect model of a vegetative state or minimal conscious state after stroke, but this is still a model where now we control very well, including with behavior, with fMRI, with EEG, the level of conscious access and conscious state, with the local global paradigm on top of it. So we started from these models in the macaques, and we inserted deep electrodes within the thalamus with several areas actually of the thalamus to test the specificity, which part of the thalamus. And to our big surprise, actually, we found that the electrical stimulation of intralaminar thalamic nuclei could suddenly induce very strong phenomena, very strong reaction in the monkey. It means that the monkey who was deeply sedated, really, with deep general anesthesia, starts having twitches with sometimes strong movement that were not seizures because we controlled with EEG. They start to have spontaneous breathing reaction, increased heart rate, as if they were wakened from the anesthesia, while they still receive in their veins a lot of propofol.
But how can we be sure that you are now not in the same position as the family members of the coma patient that opened the eyes?
Excellent question. So this is the advantage of the lab also, is to control the experiment and to add experimental tools. There are several ways. So first, we have an EEG monitoring, and we see clearly that we enhance cortical activity with thalamic DBS to a level that starts to resemble to wakefulness of the same macaque when he's completely awake and conscious. This is not enough. And here comes again our local global paradigm. That is, as you could guess, our favorite paradigm, because local global paradigm is a very good way to interrogate the conscious access of the animal during this awakening by DBS. So we performed a local global paradigm in this period of time when the animal starts to be awake. I cannot tell all the results because this is ongoing work, but I can say that's very encouraging results there.
Right. No, of course, in that sense, it's fantastic how systematic you have been, right, in building this up. This is really taking a lot of discipline and effort. This is really not easy, and I respect that a lot. But one thing that you also shared with us this morning was that you also found an explanation of why the stimulation intensity has to be so high, because interestingly enough, you could sort of only induce this sort of these reawakening signatures with high levels of stimulation of 100 hertz and above, and not with lower frequencies. So in the interlaminar nucleus or close by in the thalamus, you must be stimulating at a huge intensity to trigger the system to start to reawaken. Why is that?
Yes. So in terms of frequency, even if movement disorders, with Parkinson's, you need to be higher than 100 hertz. Then comes the intensity issue. The intensity actually will play with the activated tissue volume. So we can now model that and have an idea about which nuclei of the thalamus are involved within this stimulation. Very importantly, we control this condition. It means that with the same setting and programming parameters, with the same high intensity and high frequency, we manipulated areas of the brain that are really close on the same electrode, actually in the same condition, with absolutely no behavioral manifestation and no EEG changes. That is really key in these kind of experiments.
But how do you then explain that we need these huge intensities? So why don't you get this kind of reawakening at lower intensities of stimulation?
So in terms of frequency, for example, this is actually a general question. It's still not solved, even for Parkinson's patients who receive the same therapy, DBS, every day in many places in the world. We did some preliminary work in our models, not the anesthesia models, but in another program, and we could see that if we can we compare high frequency versus low frequency using fMRI again, which is also a favorite tool, we only higher frequency could activate cortical areas. And now in the field of DBS, there are a lot of arguments and a lot of evidence that explain that most of DBS effect is not that deep, it's superficial, it's large cortical networks activations. And it looks like you need higher frequency so that this retrograde solicitation of cortical areas happens with DBS.
And that might explain why interlaminar would be particularly well placed to do that.
Absolutely.
Okay. But on the other hand, you could also argue that what you see are just, let's say, more the brainstem systems reacting to the stimulation, and the signal travels downward towards midbrain and brainstem. And indeed, what you're looking at is a monkey that is comparable to that coma patient. So how can you exclude that interpretation?
Actually, it might very be that it also happens. However, we have evidence of cortical strong changes in cortex through EEG and through fMRI. So it would even be the best scenario that you have both reticular activating system modulation and cortical modulation.
Okay. But still the variability that you see under propofol and deep brain stimulation in the macaque, the kind of effective connectivity in cortex looks a lot more regular than in the awake state. It's not really identical.
Yes, because actually I showed very preliminary data.
Okay.
The work is ongoing, and we have also another animal. We have much more acquired data now that is being analyzed.
Okay. It's extremely interesting because maybe by driving this interlaminar system, you are reawakening cortex, but still in a more, let's say, structure-dependent way, and that's why you have these much more restricted islands of activation that you showed in your functional connectivity than a more broad connectivity you might observe under normal conditions.
You mean in non-specific way?
Yeah, exactly.
Actually,
in the resting state.
Absolutely, and we do see that if we do just resting state without any behavioral consequence, resting state on minus resting state off, we see cortical activations. However, here what we could see is the controlled cortical activation for the global effect versus the global deviant. So the global, the global deviant versus the global standard. So it's double controlled because you have within the paradigm global deviant versus global standards, and we control also. We have two conditions of stimulation at different intensities, both of them inducing cortical activations. And it shows a real specific difference for the DBS condition as compared to the other one.
Okay. Fantastic. So, Lynn, do you think that we can ever get away from using drugs to get people anesthetized?
I hope so that one day we will not use drugs to anesthetize people, because it would be much easier, because then you don't need to intubate patients, to have mechanical ventilation or to have side effects on the hemodynamic part. So I hope that it could work one day, perhaps not with DBS, because we will not implant DBS systems to the patients who go to surgery for another reason, but perhaps like systems with, I don't know, perhaps TDCS or so on, perhaps with specific.
But what do you think is a
parameters.
realistic target here? Let's say 50 years from now. Let's take a reasonable time window.
I don't know. For the time window, I have no idea.
Okay, but what do you see as the next big step?
So next big step, I think it's already understanding the consciousness better that we do now, because there's still a lot of things we don't know about consciousness and consciousness processing. For the anesthesia part, what could be reasonable is to do all the analysis we did also on EEG, because we will not put fMRI in the operating room. And then to get analysis to get like a closed loop anesthesia with anesthetic drugs that are completely the EEG pattern or the ERP patterns control the anesthetics drug delivery, and so in a closed loop manner.
Mm-hmm.
I think that it could be reasonable in a few years. There are already devices on the market who can do it based like on the spectral index, but perhaps like a better organization of this.
Okay. So Bechir, one problem I'm still having, and then we're going to the finish line, but so, okay, we start with propofol ketamine. We know what it does. We're manipulating the GABAergic system, and the GABAergic system is actually pretty fast. The time constants there are milliseconds, right? And now we try to make inferences about what this does to consciousness, also at the mechanistic level, using a measurement technique that has time constants of seconds. So we're order of magnitudes away from it. So isn't this possibly a problem? And also with the animal model you have, you could use other techniques that bring you closer to the time constants of the systems being manipulated. So first, do you see this as an obstacle that you use these techniques that are so slow that maybe you have no idea what's going on really at the mechanistic level in this brain? So how do you square that circle?
True that the fMRI scans the brain every 2 seconds, and that's obviously a different time scale. I firmly believe, like many neuroscientists, that there is no technique in neuroscience that can solve all the problems, and only multiple approaches are there to move forward. So in our case, we also have EEG monitoring of these animals, which have a quicker life time scale. But definitely our objective is to go further and to have access to the intracranial recording with the challenge to have multi-site recording simultaneously. And that's one of our next move.
Okay, right. But so you leave the option open that maybe everything we discussed so far is irrelevant because the dynamics that we have to understand is faster.
I don't think so, because take for example the intracranial electrophysiology. If you combine fMRI and electrophysiology, if you know, for example, through fMRI the most relevant sites to record, then this is clearly the way to go for. Mm-hmm. Otherwise, you will not put thousands of electrodes on the macaque brain to have access to the whole brain. Mm-hmm. And second, the dynamic resting state is now being modeled at the multi-second level. People, groups like Gustavo Deco are doing that. Of course, it's a model that is put on the fMRI signal, but it's there, and there are some interesting results.
Okay. But of course, it's also a matter of how good the predictions are of these models and how well they can be validated, right?
Absolutely.
And so on.
Absolutely.
But it's an incremental step. This is where we...
Absolutely. But I believe firmly in these models, providing that there is a mutual exchange with biological data. That's one of, let's say, my dreams, is that we are able to model, for example, the consequence of DBS, almost whatever the site in the brain. DBS is now expanding for psychiatry disorders, for consciousness disorders, on top of Parkinson's and other diseases, eating disorders. And the current paradigm in medicine and neurosurgery is mainly to rely on single observations, because one day there was a stroke here, there is a stimulation there in unwanted nuclei. So we move almost by chance. And the idea is to change completely the paradigm, is to say, well, if I have a kind of flight simulator of the brain for DBS, then this model, if it is well established, and I believe we can achieve that, then I start defining and rationalize and suggest targets for diseases in a completely rationalized manner. And here, artificial intelligence, of course, will play a big role, because it will really can say and suggest, well, for this disease, with all what we know about brain imaging in this disease, very clearly these two targets are key, or this one is probably the most efficient or the most convenient. So that's, I see it as a main way and a shift in paradigm in terms of neuromodulation and neurosurgery in the future.
Right. Very good. So Lynn, so you are now in this field also as a physician, right? On the one hand, you have these concerns about your patients, then you understand these animal models, you look at the mechanisms of anesthesia. What do you see as Lynn's law that we should follow to understand the brain?
That's a very good
Don't
question.
look at Bechir.
Yeah, that's a very good question. I'm not sure that I have a law that you have to follow. At least not for the moment. Perhaps one day. I don't know. It's a very tough question. So I think I have to.
Okay, you get a bit more time. Bechir, what's Bechir's law?
So again, for anesthesia, I think this field is
for the study of the brain in general, right? We're going macroscopic now.
Yes. Something I find it interesting from a philosophical point of view is the finding that consciousness emerge when resting state is freed from the structure. Think about we are building neural networks, machines, robots. We may start inducing some resting state activity in these circuits. What would happen if we start inducing chip configurations that are completely independent from the structure of the microprocessors and the transistors? Would we, at least at some time, assist at the emergence of an artificial consciousness?
That's a law. You see this as a law? It sounds like a prediction.
I don't know, but this would be a huge change, because if machines start to acquire consciousness, then there is a big shift, I think, in that case.
Absolutely. Does it worry you when machines?
No.
Okay. But that means what you're talking about is some sort of virtualization, right? So
absolutely
right. It's a virtualized system,
absolutely.
Virtual machine.
Absolutely.
Yeah? Okay. So then, Lynn, now that we don't have Lynn's law, which is a real failure, so we should work on that. Now, 5 years from now, I'm going to come to Paris. I'm going to visit you at Neurospin. And I want to see whether a prediction you make today was falsified or verified 5 years from now. So what's the one prediction that you feel is most important to see tested in that time frame?
I think what we want at least to test in the 5 years or the few years that will come is to see how the feed forward and the feedback works under anesthesia. Because if you use all the fMRI studies, you can make prediction about feed forward, but it doesn't give you a real answer. So the only thing that you can do is during invasive electrophysiological studies to verify this, that you have really that feed forward is present on anesthesia. That's what the assumption is today, that feedback is completely blocked. So I hope that in the next 5 years we can get answers on this.
Great. Very good. Hey, Bechir, what's your prediction? Because I'm going to find you in the same room 5 years from now.
Pleasure.
But then with another prediction. What is it?
Prediction is the following. I'm not sure we'll keep thalamic DBS as the ultimate goal of consciousness restoration. I believe the computational model would predict where should we stimulate. It would be deep, it would be superficial, it would be combined with non-invasive and invasive, and being able to build on this technology for consciousness restoration based on the computational model.
Okay, very good. Well, Lynn Uhrig, Bechir Jarraya, thank you very much for this conversation.
Thank you. Thank you very much, Paul.
The CSN podcast was produced by the Convergent Science Network of Biomimetics and Biohybrid Systems, a project funded by the European 7th Research Framework Programme. For more interviews, recorded lectures, or upcoming conferences in the field of biomimetics and biohybrid systems, go to csnnetwork.eu. And thank you for listening.